Log on, tune in, find out. Another good idea from Cambridge.
Hello everyone. Thanks for this opportunity. I'm Soyeon Shin. I work with Nicole Sorenzo at Sanger Institute. For the next 10 minutes, I'll talk about our analysis for causal inference from multiple omics datasets. This is
the microphone on.
Sorry?
It's hard to hear you.
Yeah, that's my issue.
Is it on?
I hope. Is it better? Okay. So this is the conceptual relationship of genome, transcriptome, proteome, and metabolome. The genetic information flows this direction toward phenotype. And as you can see here, each tier of organization depends on the other. So perturbation of one network can affect another. Furthermore, environment can be a crucial impact. Emerging technologies enabled us to work on these data sets. However, although for the past many years, genome-wide association studies have been successful, however, if you just focus on genome, often you will find that the effects of the variants you discovered are modest and the underlying biology is poor. To overcome these problems, our group suggested another approach, and we considered the metabolome as functional intermediates. This paper was published early this month. We conducted comprehensive investigation of genetic variants in human metabolism and discovered new insights in a range of common diseases. We identified 37 new genetic variants associated with metabolites, including nucleotide, amino acids, sugars, and others. And many of these loci match disease loci which are already associated with various diseases, including type 2 diabetes, chronic kidney dysfunctions, and blood clotting. So we believe this kind of approach could advance our knowledge of human metabolic individuality. Also, it could generate many new hypotheses in biomedical and pharmaceutical research. So keeping in that mind, integrative analysis could give us better understanding of functional relationship from multiple omics levels. In other words, we could, using those multiple levels of omics data sets, we could dissect biological and genetic determinants of normal phenotypic variation and disease states. Also, we could validate results of individual omics levels by reducing false positives caused by technical or methodological biases. However, we face various analytic challenges. First of all, these high-dimensional, highly correlated, and heterogeneous data sets they make us to work on normalization and missing value estimation. Actually, today I've heard many interesting talks, so I could actually apply some of them into our analysis. Also, since it's high-dimensional, dimension reduction is another issue. But could we really reduce the dimension biologically relevantly? Another issues in methodologies include causation, causal inference, or models including nonlinearity, because biological system is quite complex, so linearity has limitations to reveal the real underlying processes. And we'd like to distinguish causation from reverse causation or confounding factors. As you might know, multiple testing correction, validation, and replication are another issues in this kind of statistical model. especially in multiple omics dataset analysis, replication is very difficult because you have to find an independent population typed in comparable platforms. So here I introduce our project. We wanted, lipids are of the most important disease risk factors of cardiovascular diseases. And last year, a large scale of meta-analysis revealed and identified 95 loci. As a principle, we use those lipid loci. Here we aimed to find out the association sets of we wanted to find out the genetic variants associated with both lipids and metabolites. Furthermore, we wanted to use some statistical method to dissect the causal relationship of these association sets. So we find significant sets of lipid SNPs and metabolites and lipids using this significant level. And then we tested causal relationship using structural equational modeling and replicated the result. Also, the number of variables are quite a lot. We used the principal component analysis to reduce the dimension and then repeated the same analysis. I passed. The structural equation modeling is the extension of regression, as you might know, and it shares the same assumptions of linear models. Here, we tested 10 possible models where a SNP is associated with two phenotypes. This is the case of our analysis. And these 10 models were suggested by Lee et al. In 2006. We tested model 4 to model 10 because model 1 to 3 were not available because of degrees of freedom. And we compared these models to pick the best fitting model. We used the p-values of goodness of fit, and we also compared the Bayesian inference criteria because the number of parameters in each model are different. For example, model 4 shows that metabolite is the mediate of the genetic information to the lipid. On the other hand, the model 10 is the case where SNP information is just pleiotropic. These assumptions and structural equation modeling has advantages over other statistical models. For example, Mendelian randomization, the classical Mendelian randomization has has hypotheses and assumptions, but they are not flexible. For example, they do not allow direct and indirect effects. Also, Bayesian network analysis or its application kind of limits the estimation in two steps. So first, it estimates the effect as considering the variable as predictor, and then in the next step, the other way around. The first response and then predictor. On the other hand, the structural equation modeling can estimate them simultaneously. However, this is statistical model and it's based on linearity, so it cannot detect nonlinearity. Also, hidden confounders and measurement errors can mislead causal inferences. However, this advantage can be shared in any other statistical model, and it's also the issue in biological experiments. So in our analysis, we tested 95 lipid loci associated with serum lipid levels, and we applied SAM to test causal inference. And almost those 7 models that I showed in the previous slide, we found that 260 association sets show that model 4 is the only significant or the best fitting model. So it really showed that these metabolites in these associations are the mediators of this model from lipid SNPs to the lipid. And we found them in 3 loci, including FAS1, GCKR, and ApoA1. Principal components also showed the similar results in 2 of those 3 loci. So we suggest that SAM is an appropriate statistical instrument to dissect the contribution of intermediate phenotypes to complex biological pathways. So we'd like to keep the SAM analysis and then extend our analysis now from metabolome only to the transcriptome as well. I think somebody in the early morning session mentioned the mother data, which were gene expression data sets of twins UK cohort population. data set. And we also have the same data set. So genome, so we have genome, we have GWAS data set and gene expression data sets and metabolome data set. These are the number of variables, and overlapping sample size of 600 have all of these data sets available. This is we are very early stage of this project, so I'll just show you how we are dealing with missing values and dimension reduction. We are really welcoming any kind of advice for our analysis plan. So missing value is an issue in multivariate analysis. You can either ignore, but it reduces the sample size and water waste. So we have to impute them. Usually, simply you can use either row mean or you can use other methods. And in these two papers, they say in their simulation studies, they show that Bayesian principal component analysis is very efficient in imputing missing values. So we tried it using the R package PCA methods, and this Bayesian principal component analysis is actually written for the missing value imputation, so it does not really capture the final scores, does not capture the variance of the whole data set. So we have to validate our results. Assuming that we already imputed all the missing values, then we proceeded to dimension reduction. If you have only one data set, then the first method coming into your mind is principal component analysis, but it's limited to linearity only. So you can either use Conner method, Conner PCA, or multidimensional scaling, or like those methods are available. But when you want to reduce your dimension in two data sets, what you can consider is canonical correlation analysis. So you reduce the dimension while maximizing the correlation of two data sets. And also partial least squares can be applied. So we tested regularized canonical correlation analysis because we have a high dimensional data set. And as you can see, the transcriptome and metabolome itself has some correlation amongst those variable sets. And however, the cross correlation table here does not show any interesting signals. So after applying regularized CCA analysis, as you can see here, the latent variable are orthogonal to each other were captured, and it shows that there's no correlation going on except for the diagonal line. Also, in a cross correlation matrix shows that the first component of the X and the first component of Y are still keeping the correlation, and this is the bar plot of correlation of cross correlation sets. We are still exploring many methods because open questions remain how best to integrate the multiple omics data sets to understand underlying biological mechanism and infer causal pathways. And as I mentioned earlier, we'd like to get advices, and we'd like to collaborate if you have a better idea of how to work on this kind of analysis. Thank you.
A question for later, because we have one more slide before the coffee. Try to handy. But let's thank So Yoon again.